All right, terrific. Thank you for joining today. This is going to be recorded, and I take it, Chad, you have gone ahead and start the recording.
Yes, sir.
Excellent. So it will be available off line in the very near future. But thank you for showing up, and we expect others to join during the presentation. So this is about direct access to the microbial dark matter for AI-driven polypharmacology and drug discovery. It really is focusing on gold-in data solves AI's garbage out problem, saving $1 billion per drug. Now, that's a bold claim. We hope the following presentation will help support that and help understand where there can be a shift. So the shifting strategy, de-risking the asset from phase 2 in the clinic to lead stage in preclinical for lower risk and a substantially higher ROI. So what we're talking about there is many investors, many pharma companies back around 2002, 2004 basically shifted their strategy to only investing in de-risk asset. Sometimes that was at IND, sometimes phase 1 or phase 2, and that made perfect sense. What we're here to say is with this opportunity that's in front of them, the ability to shift back to earlier and have a much higher ROI and a much higher probability is what we're also going to talk about today. And we think that will lead to the upcoming platinum age of pharma. Now, in front of you as you were waiting were a number of quotes, all discussing essentially the lack of data or not having the right data. That is a key thing that is realized, call it a key theme that is realized by many that have been pursuing drugs for decades. It's not just about the right data. It's also about accessing nature's drug-like small molecules. So over the decades, the costs have gone up. The real productivity has gone down per cost. This is called Eroom's law, which is the opposite of Moore's law. And we'll talk a little bit more about Eroom's law in a second. But there's certainly a discovery gap. Current methods rely on limited libraries, actually limited real libraries, and then they rely on synthetic libraries that are extensive and dilutive. And we'll talk about that in a second as well. The high failure rates, the lack of natural molecule priors or diversity results in costly failures at drug development. So we'll talk a little bit more about that. So no access to nature's best molecules. When 50% of all the drugs on the shelf came directly, indirectly, or inspirationally from microbes. And that 99% of that source is unobtainable and untapped. And we'll talk a little bit more about that as well. So what is Eroom's law? And why is it really important? And why are some of you hearing about it for the very first time? It's just the recognition that productivity in drug R&D is falling, not rising. This metric, for every 1 billion spent in R&D, we get half as many approved drugs every 9 years. Like I said, it's the reversal to Moore's law. Spelled backward, that's where Eroom's law came up. So developing new medicines requires vastly more money and time, making breakthroughs increasingly difficult and costly. Well, all those that have studied Eroom's law have come up with certain things. Well, the low-hanging fruit is gone. The easiest diseases to treat have already been addressed. Incremental benefits, newer drugs, often only modest improvements. regulatory hurdles, stricter safety standards, driven by past drug failures, make it harder and costlier. Flawed models, reliance on outdated animal models and brute force. Well, maybe some of that is true, but we're actually saying it's the wrong molecules and it's diluting the training. It's a little simpler, and we think we can prove this. So there is a paper out there. It's in pre-publication, so it hasn't been peer-reviewed yet, but it's called Reversing Eroom's Law, the Finitude and Dilution of Small Molecule Discovery. Um, we'll make sure that that link to that paper is in the notes, and it is something that can be found pretty easily on Google Scholar as well. So let's talk a little bit about that. This is the mathematical proof that essentially we're going about AI and drug discovery wrong. Theorem number one is nature's finite playbook or saturation. So nature uses a set of limited rules, finite grammar. A lot of those we found, even though we've only explored less than 1% of nature's true rules. But we certainly understand a lot of structures, and there's some structures left to be found. But it's the details, it's that grammar, that communication that we're missing. So right now, we know we're going to run into a wall with nature's knowledge. And we've run partially into that wall, but that wall is still a long way away. That's theorem number one. Theorem number two is the dilution, the synthetic dilution of the infinite space. So what we're talking about there is in the infinite space, you could have virtually any kind of drug-like molecule that looks good but doesn't work. In fact, the finite space is relatively small. And if you think that the finite space in nature is relatively small, it really has to be put in perspective to the infinite possibilities that exist. So right now, if you think about the infinite possibilities, we as drug discovery world have found trillions of potential molecules and are adding trillions potentially to that through computation and AI. But even with trillions and trillions of molecules potentially to play with that are mostly unreal and won't work, the reality is that's only zero point 0 followed by 100 zeros, 1% of the possibilities. So you can play in this infinite space and dilute training sets and really not get anywhere. And that's part of the problem. You've got the finite space problem, you've got the infinite space problem. So theorem three, mathematically again, is the hybrid solution. Taking the finite priors and expanding finite priors essentially so that you can train the infinite space. Now that's important, and we'll try to explain that a little bit more. The math is a little complex when you look at it, but anybody can drop that paper into large language models and ultimately ask questions till you're blue in the face. So I recommend downloading the paper, reading it, and then throwing it into AI and saying, explain this to me deeper. Why does this work? How come we're stalled on finding finites? And what does it mean if we find more finites? How does that improve the system? So there is massive data generation strategies that are actively contributing to the problem. the dilution problem by expanding the infinite faster than the finite prior discovery can harmonize the data. What this is really saying is we could endlessly explode the bad side of data and ultimately get nowhere. And that's what we've done. And that's what we have even done within the NIH, ARPA-H, DARPA's biotechnology program. We have invested heavily in expanding data that is irrelevant. That's part of the problem. So when you look at this and you look at the model and you look at the paper, it is really confirms that we're exponentially the counter case right now. That's the infinite dilution portion of the paper. It says finite, but it should have said infinite. Sorry about that. So lambda in the paper is growing at millions of experiments per week, and upsilon, the search space, is growing by trillions of molecules. The result, the infinite is expanding faster than tools can actually filter it. We have petabytes of data, but very little of that is good for AI. We're prioritizing volume and speed of data creation over the value and relevance of the natural priors. This is a prime example because this $3 billion doesn't count the hundreds of billions of dollars that was spent previously. But just recently, 4 companies, billions of dollars to expand what is truly the infinite data space. It's not expanding the finite priors that you use to train the data. And that's important. These are not real molecules. This did not come from nature. These molecules are not used as cellular signaling molecules. That database is actually small right now. And even that database is convoluted. That's why it needs to be essentially curated to be the best database possible. So where does that data need to come from? Well, historically, as we talked about earlier, 50% of the drugs on the shelf came from microbes. Even though that has been a hotly explored space, we've only tapped into less than 99%. So, I mean, we've only tapped less than 1%. So there's the other 99% that's available. So the right source is well documented throughout the history of research. And anyone can look that up on a large language model. It is absolutely declared as the best drug potential source that exists. We just don't have access to it, or we believe we don't have access to it. But looking at it genomically, there is proof that the overlap to the human microbiome is huge, which presents the opportunity to understand what's going on in the human in a way we can't right now. So Biosortia created a technology, and this has been a technology that's been in research and development phase for 15 years. It's called industrial scale microbiome mining. And it goes out to the microbial world and collects massive samples without damaging those samples, without changing the metabolic pathways, preserving them almost immediately from their source habitat in a very environmentally friendly way in order to obtain those natural priors to perfectly align with AI, basically providing the training sets the drug discovery has never had. Now, what does it really mean when you go to that microbial world? There's been studies showing that currently natural products beat synthetic products, but there's never been a focused understanding of microbial starting points in comparison to synthetic starting points. So being able to extrapolate the data that does exist in peer-reviewed scientific papers, what we see here is you must start 40 synthetic programs to get to one approved program. And this waterfall is incredibly costly because that means 40 programs at preclinical, 20 at preclinical to phase one, 10 phase one to phase two, and et cetera, putting out a cost of $1.49 million for a synthetic program to get to one approved drug. But when you look at the history, and this isn't projection, this is pulling out history and extrapolated by published papers that are referenced in this paper, you got to start 4.4 programs to get to one approved, which means you spend a fraction at lead to preclinical, preclinical to phase one to phase two to phase three. And then it starts to equalize as you get closer to that finish point, which is an approved drug. So you're only spending 4.46 million versus the 1.49 million, or that billion-dollar difference. That's going to shift dramatically the opportunity to decide when to invest, and we'll talk about that a little later, and how to maintain the highest return on investment. So the dark matter of the microbiology world, what is being brought to light by industrial scale microbiome mining, is basically the gold in, gold out theory. 4 billion years of evolutionarily optimized microbial chemistry that not only works cell to cell, but works with inside hosts like humans. Our data sets are expanding dramatically faster than real training sets outside of industrial scale microbiome mining. That is allowing us to review more real molecules that have ever existed and projecting to be well more than 1 million molecules within 3 years, with training sets that get expanded to be able to do those predictions. So that'll allow us to find the high value leads now. But ultimately, that will allow de novo design at the highest level. So when you look at what Biosortia is doing, we're looking for the microbiomes that have those hidden metabolites, those hidden small molecules that can end up informing the finite side of the equation with the language that exists for the molecules themselves. To do that, we've got to recover a massive quantity of microbiome, process it effectively. Now, when you talk about our scale, we're talking about 8 Olympic-sized swimming pools and protecting that biomass and preserving that biomass without lysing and without metabolic pathways changing. And ultimately, we can take that into our early machine learning and our large language models to be able to understand what these molecules are. And we have 2 approaches. We can pull the molecules out directly for direct screening, or we can prioritize with AI. And this gives us a closed loop self-improvement system. Essentially, like the last slide showed, we scout and find nature's best small molecules. We process it, we analyze it, we predict the SMILE strings, which allows us to predict derivatives, to predict the ADME and the tox, to predict the activity, all of this in silico, then prioritize and either directly obtain from the biomass or synthesize. And again, The 4.4 programs to get to one approved drug was prior to AI de-risking with accurate training sets. So we actually expect in the very near future to get to a point early and to know early that if you start with the right de-risk molecules at preclinical, at that lead stage, you will be able to get two programs started and one molecule, one approval. That's a massive cost reduction. And that's what we believe this technology will be able to allow. Now, the nice part is execution itself allows this continuous improvement cycle as the data comes back in to the system to learn and predict and improve. Ultimately, de novo design will be so precise coming out of this system of understanding the language of nature that will be in a position to target appropriately with molecules that are designed using nature's language. Now, what does that really mean for the investment side, whether it's pharma, whether it's a biotech, whether it's a venture capital firm? What it means is a dramatic shift because essentially the investment has been once the risk subsided and you had a higher probability. Some of that risk occurred probably at a high risk stage in phase one or preclinical IND previously. So if you look at the blue lines, that's the old order. But they were making bets pretty early. The safe place to make those bets are phase two to phase three. And that shows you where you've got a much higher probability of return on investment. What we're seeing because of the cost reduction here is that the bets can be made back at the lead stage, depending again on what the value of the molecule should be able to return. So if it's a lower value, maybe $1 billion a year, you could wait till you get into the preclinical to phase one stage, not at lead. But there's going to be some programs where the economics justifies throwing down the investment bet early because it'll cost less and your opportunity to return a much higher ROI is supported by the math. So let's talk about a proof of concept. Let's talk about potentially a paradigm shift in weight management. So GLP-1 drugs aren't the final answer. They were the first answer. GLP hits that target hard, and as it hits it hard, people are searching for better options, more tolerable, designed for long-term success without drawbacks. And that's where we believe the answer lies in the biological dark matter and using the system we've created. So I don't have to explain to anybody that the weight loss market is huge and growing, and we're basically down here. So there is going to be a lot of additional investments in the weight loss market. There is no doubt about that. What we have found using our technology, using the natural priors to improve the predictability of unknown novel molecules and the structures of unknown novel molecules, and then using the best of in silico de-risking, we found a molecule that actually hits not only GLP-1, but it hits other key targets. Basically, it will intelligently reestablish the body's natural stop signal with a single oral molecule and ultimately hit multiple targets, a little more harmonizing to hitting those targets. And this gives a little better understanding of what we're looking at here. And that is that the single compound, which is under 500 molecular weights, not only hits the allosteric modulation of GLP-1, but it hits the MC3R, MC4R, and it hits other areas. So we like to represent this in the hypothesis that essentially imagine you've had the best meal of your life, you're full, you're happy, you're satiated, and if you decided to take another bite, you might actually feel sick. That sickness is on target. That happiness is on target. This is what it looks like nature made in order to attack this target. Now, what do I mean by attack this target? From looking at large language models to try to explain where this molecule could have come from, it appears this molecule might be 1.5 billion years old, plus or minus hundreds of millions. The reality of it is it looks like it used its signal or that molecule to convince its competitors they didn't need to consume its resources. So instead of being an antibiotic that killed off its competitor with toxicity, it basically convinced it, you don't need to eat here. Well, ultimately, higher animals adopted that into the endocrine system. We believe that's the hypothesis. And how that would work in a human is that microbe, which would inhabit the human microbiome, is trying to protect its habitat. It wants the resources, it wants you to bring them in, but it doesn't want to inundate its habitat. So it sends those signals out, stop eating. That's where these signals came from. The microbial world potentially could be the controlling factor in weight loss, ultimately through the signaling. Now, what happened to that signaling? Where did that signaling go? That signaling between antibiotics, Western diets, overeating could have inundated that microbe so its signaling is muted or no longer existent. It could have been outcompeted by competing microbes that took over its space. So that signal could be gone. We don't believe it's a microbe to reestablish that. We believe it's the molecule to reestablish that. That's what we think is the best approach forward. So this asset is part of a portfolio. The way Biosortia has been able to target potentially opportunistic areas like weight loss or other areas like antidepressant, anti-anxiety from the gut rather than from the brain. We're looking at how to use polypharmacology in order to attack what is the target or the range of targets that exist. So in this particular case of our BIOS 1 library, we found another 38 novel scaffolds that relate to weight loss. So the recognition that the microbial world is rich in sources of target hitting molecules in polypharmacology without promiscuity is one of the things that we're seeing right now. And that is expanding our ability to be able to support somebody's pipeline earlier at a lower risk, lower cost, with a higher probability and a higher ROI. The next step on this molecule is we're going to obtain it either through synthesis, which it is got that synthetic amine ability to it. It's a novel scaffold and structure that doesn't exist in any library today. So we were able to file 3 patents on it. A fourth one is getting ready to be filed. And ultimately, we were able to do a Merkesh formula on this entire portfolio that represents the lead. And that represents more than 2,000 molecules that are tunable. So we're building IP around what we've been able to learn to support this weight loss asset. So, in summary, natural priors that come from nature, specifically microbes, because microbes are the most connected drug-like molecule source that exists. And in fact, a lot of plant molecules came from microbes, either directly or through gene transfer of the genes of the microbes. So the reality of it is a lot of natural products came from microbes, but definitely not all of them. So the microbes create the best multiple shots on goal, the potential best in class dual action polypharmacology, enabling, we believe, this next spurt of growth in the pharma world, the platinum age of pharma. We believe microbes drove the first one. Microbes, because we have got to understand them better than we have ever had in the past, will drive the next platinum age. So we invite anybody to reach out to us, whether they're a pharma company that is looking to expand their pipeline and has target areas they'd like to try to find molecules for, whether they're a biotech with a single product and may want to have a backup opportunity, whether they're asset accumulators, people that invest in IP. We think that thousands of new IP will come out of this approach. And accredited investors are always welcome to give us a conversation at any time. Give us a ring. I'm going to stop there. And Chad, I think, is going to coordinate any questions that may have come in.
Ross, we don't, the chat is open. A couple people definitely want the presentation afterwards, but I think everybody should be able to unmute themselves right now, so we can have some conversations that way also.
Does anybody have any questions? And you're able to unmute yourself if you do have a question.
I have something to ask.
Marcia, you are welcome to.
Okay, so obviously this is super technical. I am kind of geeking out on some of the things you say, but I'd like to really to get like this is the one side of it, perhaps it's from the investor side. I'd like to get a clearer picture of what that would really look like by way of as a competitor to GLP-1. Is it a therapy drug? Is it a drug, or is it where does it stand? Is it a prescription drug? Like how at the end result, what are we really comparing? How does that work? Does that make sense?
It does. And it's at its earliest stage right now, which is computationally de-risk with a number of molecules that are worth taking into the wet lab and exploring. And that's what we're going to do next. It will go through the FDA process. It will ultimately, the best outcome is getting an approval, being a oral drug, maybe the size of a baby aspirin, that someone could take once a day in order to really help them. Could it be as many as 3 times a day before meals? Possibly. All of that has to be uncovered through the preclinical and the development itself. But there appears to be a generous amount of opportunities around this molecule because its structure seems tunable. But we would believe it would be a drug primarily. You never know what goes into, the status of being over the counter some year in the future. That may be 50 years. It may be never. All of this is so speculative. That's why it's got to start down that process. But we believe that since it's a microbial molecule, since it's been computationally de-risked, There is a series of structures. There is opportunity to reduce its risk through derivatization. We believe it has a solid opportunity to compete as a first class competitor against any other weight loss approach, including GLP on its own, hitting it hard, or anything else that somebody may come up with. We think it's the best shot that we've seen, obviously. We don't know what everyone else has seen, but we've seen to potentially be a drug that hits the market and has an impact.
Okay. And so at this stage of where it's, when you say derivative, let me back up for just a second for the derivative side of it. Uh, are you suggesting that there'd be other types of therapies that would fall under that sort, that would compete against something else other than GLP-1?
Well, there is a number of people that have been pursuing various opportunities around weight loss. The MC4R, MC3R has been looked at for over 20 years, trying to combine it. Right now, the GLP-1s are generally larger molecules that need to be injected. So this is already potentially a game changer, especially since we believe this molecule came from a microbiome. It was
That's interesting.
which
Yes.
which it makes it orally available, and its structure allows it to withstand, let's say, making it through the stomach and the stomach acid and getting into the rest of the digestive system to have its impact. So uh,
Um,
we think it's a great starting point, and we're pursuing it, and only data and time will tell.
Okay. And when you say you've de-risked it, you're speaking specifically financially, right?
No,
Oh,
computationally. So
computationally. Ah, okay.
yeah, everyone in the world has tools available to look at its ADME, to look at toxicity, to look at its activity, to look at its targeting. Is it on target? Is it off target? So we've done, we believe, enough preliminary work on it to justify making the molecule and testing it in the wet lab and in early animal models.
So something that's very interesting that you just said, between large and small molecule. I don't know if everybody here on the group already understands that. And I'm coming to understand it, over time with you. But I'd like to, if you could kind of separate, how that, because you're saying there's the actual molecule and its derivatives. So are the derivatives the ones that are the smaller molecules that will make it easier to over the counter, or make it, so you don't have to take it as an injection, and the larger or is the whole thing all under, are you understanding my question?
I do. So let me try to explain what a derivative is. You hear a lot of, and I'm going to use this example, and I hate using this example. Um, when you look at the cannabinoid and the cannabis marketplace, and
Yes.
you look at the main ingredient that creates the activity of feeling high, um, that's a very well-known molecule, but people go out there and change it slightly.
Hmm.
That's a derivative of the known molecule. By changing it slightly, they may not actually be changing the activity, but they may be creating a much higher risk for whoever takes it. So you hear a lot of foreign groups that create a high in a vape and then try to sell it in gas stations in the US, and the FDA and
That's
everybody runs around trying to stop them from doing that. That's
yeah.
actually, they've derivatized that key
That's
molecule.
yeah. I,
And
I
and
understand why you wouldn't want to talk about this, but that's exciting to know that that's what you mean, because that's a huge market in general, right? I mean, it
well,
is. I used to live in
well,
Holland, so
we would attempt to cover all versions of that in the IP and have that covered so that nobody could do that. That
Okay.
would be the point. The whole goal and the objective is to understand what derivatives are there, what they can do. And this looks like it
Yeah.
is a molecule that, like many molecules, could have variations, and the IP needs to cover those variations. And we're thinking ahead.
And that would mean, to some extent, am I thinking in the right way that that would mean, let's say, for an investor's perspective, that they could white label something from one of the derivatives that would be specific to them and what
Well, it
they
depends
do?
on who ends up owning the patents. So
Okay,
that is a long way down the road. The
I
important
understand.
part is Biosortia and whoever partners with us on this weight loss molecule, whether it's investors, whether it's a pharma company, the whole goal is to put it in a box so it can retain value, not dilute value. And that's what everyone involved will be working for, is to make sure that we create the best product for the marketplace that has the best positive impact on those who need this.
Okay. I understand what you're saying. It's helpful to know though, because if we're talking to investors from my side, it's a whole entire package, right? It's the IP and its derivatives and the prospects of what it can be and be done with, right?
Yeah, and we can talk specifically about that offline when you talk about investors that you're talking to and trying to introduce to this opportunity. So we'll talk
Okay.
outside the public. Any other questions? All right. Well, I think we'll wrap the recording and make it available. And obviously,